I started sharing the screen. Can you see it?
Yes, it's visible.
Yes, sir.
Yes, sir.
So is it fine if I start the recording?
Mm, sure, maybe after a few minutes, because it'll, once I start, I'll let you know, and just before that, and you can start then.
Okay, sir.
So let me know when you are ready to begin.
Yes, sir, sure, sir. Just one more minute and then we'll start. 1 or 2 more minutes. So we are actually also streaming on the on YouTube. So once we go live there, we can start. Fine, sir?
Yeah.
Okay. Okay, sir. Good evening, everyone. And thank you, sir, for joining this talk. Welcome to the second talk of Astrobiology talk series, arranged and coordinated by Parsec and ESI, clubs from IISER-TVM. It is great pleasure and honor I announce the speaker, Dr. Supratim Sengupta. He has completed his master's from IIT Kharagpur in the field of physics. He went to Institute of Physics, Bhubhaneshwar to complete his Ph.D., followed by his first postdoc from Theoretical Physics Institute, University of Alberta. Currently, he is a professor at IISER Kolkata. From the description, you can already see that he is a physics person. And so this talk is going to be a physicist perspective on evolution of biology, life from chemistry. Before we begin, I would like to request everyone to keep their microphone off and the videos off. And towards the end, we'll have a Q&A session. So whoever has questions can post them in the chat box. I'll read them out for you at the end. Or if you're comfortable enough, you can switch on your microphone and ask the question yourself. So now I would like sir to take it on from here. Thank you, sir.
Sure. Thank you. So thanks for the invitation to give this talk. It is indeed gratifying to find out that a group of students who are interested in astrophysics is also interested in issues pertaining to the origin of life. And as you found out, I'm a physicist by training, but the perspective that I'm going to provide here in this presentation is certainly not the only perspective on the topic of origin of life. Because of the highly interdisciplinary nature of the subject, people from different research backgrounds, including biology, chemistry, geology, as well as physics, have all made seminal contributions to this field. From a chemist's point of view, they are primarily interested in figuring out what are the basic building blocks and how are they synthesized, the basic building blocks being the nucleotides ATGC for DNA, or if you think of RNA, they are AUGC, as well as the amino acids. So from the perspective of chemistry, the origin of life, issue of origin of life is also related to the issue of how these basic building blocks, the nucleotides and the amino acids, were synthesized in the absence of enzymes. Biologists bring a somewhat different perspective. They are interested in an evolutionary perspective, and they want to find out how chemical evolution eventually led to the emergence of a self-replicating organism, which may have looked very different from the organisms that we are familiar with today. Geologists are, on the other hand, interested in figuring out the correct environmental conditions that are necessary for life to originate. And keep in mind that we are thinking of a scenario which took place about 3.8 to 4 billion years ago. So it's extremely difficult to make precise predictions on what the exact environmental conditions were that facilitated the emergence of life. But one can make plausible guesses and try to collect evidence to either falsify or verify those kinds of guesses about the right environmental conditions. And so geologists are involved in trying to find out the right environmental niches which facilitated the emergence of life. As a physicist, I'm interested in developing mathematical and computational models to understand the process of origin of life. And I must add that doing so provides a very novel perspective, because many of these questions require experiments which are extremely hard to perform. Not only that, there's a lot of uncertainty about the precise environmental conditions, the precise prebiotic chemistry and biology that went on about 4 billion years ago. And mathematical and computational models can really help in identifying plausible scenarios that can supplement the work of experimentalists, whether they be biochemists or geologists. And they can also sometimes point the experimentalists in the right direction by identifying scenarios which seem plausible and which can then be explored in greater detail through biochemical experiments in the right environmental niches. So to start with, I should pose the question, what is life? If we want to understand the origin of life, we need to figure out the definition of life. There was a lot of controversy about this question till some time ago, when a biochemist called Gerald Joyce, who was working at the Scripps Institute, came up with this definition, which now seems to be accepted by a large community of researchers. So according to him, life is a self-sustained chemical system capable of undergoing Darwinian evolution. Now, as you can see, the definition is extremely broad in scope, but we need to break it down into pieces to understand what it really tells us about a living organism. So what do we mean by Darwinian evolution? So think of a population of entities which are self-replicating autonomous agents, shown in red. They can reproduce and pass on their genetic information to their progeny, but replication inevitably is error-prone. And during the process of replication, mistakes are made, which leads to production of new variants. And eventually you can see that due to evolution and the process of error-prone replication gives rise to these variants which are represented in different colors. And one of the variants represented in green eventually start proliferating because it happens to be better adapted to the environmental niche where this population is growing and evolving, and hence it is selected more frequently compared to others, as a result of which its frequency in the population increases. And so this is what Darwinian evolution is all about: error-prone replication accompanied by selection on variants, which leads to proliferation of better adapted variants, right? So life then is, according to this definition, is a self-sustained chemical system, but it leaves a lot to be desired because life as we know it is much more constrained than this definition would suggest. For instance, all the living organisms that we know of, starting from the simplest bacterial cell, is enclosed within a protective cell wall, right? And they are carbon-based, which uses DNA for storing genetic information, and synthesizes proteins, which then perform crucial tasks, including tasks involving replication and transmission of hereditary information to progeny. And protein synthesis uses a genetic code which encodes only 20 amino acids. So despite the extremely broad scope of Joyce's definition of life, life as we know it is much more constrained in this following manner. So when we come to the question of origin of life, one of the plausible hypotheses that was put forward several decades ago is called the Rna World Hypothesis, which posits that life, unlike the kind of life we see today, which is based on DNA and proteins, did not start in that form. It started with RNA molecules only. And one of the motivations for proposing this RNA world hypothesis of the origin of life came from the fact that RNA was discovered to be capable of performing many of the same functions that are carried out by DNA and proteins in the current scenario. So we know that DNA is very efficient for information storage. It can catalyze reactions, and it can regulate expressions of various genes inside an organism. And over the last few decades, the RNA analogs of these have also been discovered. RNA, like DNA, is also capable of storing genetic information. It can catalyze reactions because RNA enzymes like ribozymes were discovered, which were effective in catalyzing certain chemical reactions, including self-cleavage and replication and so on. And RNA was also found to be capable of regulating gene expression independent of proteins. And this was found out when RNA regulators like riboswitches were discovered about 20 years ago. So given the fact that RNA can perform many of the same functions that a DNA protein world can exhibit, it seems plausible that life arose in the form of RNA first, where the function of information storage, catalysis, and regulation were all carried out by different forms of RNA. So this was the motivation behind the proposal of the RNA world hypothesis. But the evidence was circumstantial, as you can guess. We do not yet have any direct evidence of any organism that is based entirely on RNA. So in order to understand the emergence of the RNA world and validate the hypothesis of the RNA world as a plausible hypothesis for the origin of life, we need to do a lot more. So we need to understand how non-enzymatic chemical reactions led to the synthesis of biopolymers like RNA, right? So when you go back about 4 billion years ago, the chemistry was very different. You did not have enzymes because most of the enzymes are created inside living cells. It is not even clear how the basic building blocks like the nucleotides A, U, G, C were synthesized. It's only recently in the past 10 years that chemists have found out ways of synthesizing these nucleotides without requiring enzymes. And we also don't know the precise nature of the environment in which these RNA polymers can form, the polymers that would be required to kickstart life in an RNA world. So the emergence of RNA world requires several steps. It requires the basic building blocks to be synthesized in the absence of enzymes. It requires non-enzymatic chemical reactions in suitable environments to bring together these building blocks to create polymers like RNA, which are capable of storing information. And these polymers, long chains of AUGC, must also eventually lead to the formation of RNA enzymes, which can catalyze reactions much more effectively. So one of the questions that are associated with the origin of life is, how did the first ribozymes emerge in the absence of catalysts? Right? And the key aspect of this question can be encapsulated in the figure shown in the last row here. So it's not enough to say that we know how to build the components that lead to the RNA world. The components being the nucleotides AUGC, RNA polymers, where the nucleotides can polymerize and form long chains. And some of these long chains will acquire catalytic function, which allows it to act as catalysts to speed up certain reactions, which are essential for RNA world to function. And we also need lipids because if we think of the RNA world, we also need to think of a key aspect of life, which is it is enclosed in a compartment, and compartments are typically formed out of lipid vesicles. And so one of the key questions regarding origin of life has to do with how all these components, even though they can be synthesized independently, how they come together in the form of a protocell and produce a self-sustaining chemical system which is capable of undergoing Darwinian evolution. The second aspect of the origin of life is to understand how the transition from the RNA world to the DNA protein world took place. And one of the steps that facilitated this transition was the process of the development of this language of genes. And I will come to that in the second half of the talk. So in the first part of the talk, I'm going to try and focus on addressing how some of the key components of the RNA world, namely ribozymes, were synthesized without enzymes. And there are essentially two ways of doing that. You can have a linear strand of RNA which can act as a template, and nucleotides can get attached across the corresponding Watson-Crick forming Watson-Crick pairs across the corresponding nucleotides in the template. So you can see from the second figure here, these nucleotide U forming a corresponding AU pair, then you have a C which forms a GC pair and so on. And the top one is called the template and the bottom one is called a primer. The primer gets extended as nucleotides are added across the nucleotides on the template, and this produces an effective way in which a sequence which is complementary to the template can be produced. But there's also another way of producing long chains of nucleotides that lead to an RNA polymer, and that is by a simple non-templated concatenation process, which you can see in the first figure. You can see that starting from a single nucleotide, other nucleotides can attach to the end of the existing nucleotide to form a dimer and then a trimer and so on. And this process can go on for a long time to produce long polymers. But a key aspect of these processes is how efficient they are in producing long polymers, right? And as you can see, for a concatenation reaction, which takes place without the help of a template, the reactions are quite slow. So when nucleotides keep adding one after another to an existing chain, making that chain grow longer, the typical rates are of the order of 0.6 per hour. And all these reactions have to take place in the absence of enzymes, because we are talking about an epoch where enzymes were not yet synthesized. This is, in fact, one of the processes by which RNA enzymes can be synthesized, but there were no enzymes to speed up these processes. So why do we need to consider both non-templated concatenation processes as well as templated concatenation processes? And the reason has to do with the rates at which these processes occur. And you can see from the second figure, the templated concatenation process occurs at a much faster rate because which has been measured in the lab by various groups. But this process of forming a new RNA polymer which is complementary to the template is error prone, because during the process of elongation, by mistake, a non-Watson-Crick pair can be produced when a wrong nucleotide gets attached to the leading end of the primer. And this is shown in the last figure here, where a nucleotide C, which is shown in red, has gotten attached to the end of the primer, to the G nucleotide here, but then it has formed a bond with the nucleotide A in the template, which is not a Watson-Crick pair. So the template-directed replication process, which produces an RNA polymer, which is complementary to the template, is an error-prone process. And when a mismatch occurs, it slows down the subsequent addition of nucleotides on the primer and slows down the process of primer extension. So this is one of the bottlenecks that one has to contend with in order to understand how long polymers can be produced or how difficult they are to synthesize in the absence of enzymes. So in this slide, I was focusing on ways in which polymers can be synthesized, either through non-templated concatenation reactions due to addition of nucleotides to the end of an existing polymer chain, or through template-directed processes which occur at much faster rates, but which are error-prone, and therefore it leads to sequences which are not exact complements of the template. But these error-prone replication processes also are useful in the sense that they lead to new variants being created, right? But there's another process that we have to keep in mind, and that is the process by which these polymer chains can break apart. And that process is called the RNA hydrolysis. So hydrolysis leads to breaking up of an existing polymer chain at specific locations. The locations can be random, but those locations which are paired are better protected against hydrolysis than locations which are unpaired. So you can see from this figure, there are some paired regions of the sequence because of bonds which have formed across the template primer pair, but the rest of the template has a dangling end because there are no paired nucleotides which have formed phosphodiester bonds with that region. And so hydrolysis is more prone to attack the bonds in the unprotected region, and less prone to attack bonds in regions which are protected because of existing bonds in either side of it. And so there's another region which can form in an RNA molecule, which corresponds to a loop here. You can see the nucleotides being shown in blue, and these are semi-protected. They are in the sense that they don't have paired nucleotides, but they have bonds which form with nucleotides before and after them. And so they are semi-protected compared to the dangling portion. And these are also somewhat less prone to hydrolysis than the nucleotides, than the bonds in the dangling portion. So these are the processes which compete with each other. The concatenation and template-directed primer extension process tries to increase the length of polymers, whereas hydrolysis process tries to break up existing polymers into smaller beads. And these two competing processes determine the nature of the polymers produced. And one of the key criteria for producing enzymes or any molecules which have a function is that it has to be a long molecule, right? So one of the challenges has to do with the competition between the process by which polymer chains are extended and the process by which they are broken up due to hydrolysis. And these two competing forces will eventually determine whether it is at all possible to form functional biomolecules which can fold into complex structures that are necessary to perform certain biological functions. So as I said at the beginning, we as physicists are interested in developing mathematical and computational models to understand the processes which lead to the formation of these different components that make up the RNA world. But one of the key elements is the environment. So we don't really know for sure what the right environmental condition was that led to the formation of such long RNA polymers, which eventually evolved to be capable of performing certain functions like catalysis. So this slide brings me to that key aspect of the problem, which is to figure out the right environmental conditions. And there has been a lot of work in this area, and the consensus, at least for a certain group of scientists, is that the condition that is most suitable, or at least a plausible one, for the emergence of life existed around volcanic pools in primordial Earth. And these conditions, so these volcanic pools were essentially pools of heated water with a lot of minerals, which would undergo phases of hydration and rehydration and dehydration. So in the morning, when the sun was shining brightly, the pools at the edge of these at the edge of the pools would start drying up, and later on at night, when it would become cold and wet, it would start getting rehydrated because of rainfall, which was quite prevalent in primordial Earth about 4 billion years ago. So why do we need this kind of rehydration and dehydration cycles? So keep in mind that for a polymer to extend in length, we need monomers available in the environment, and the monomers should be able to move around, and the reaction which leads to the concatenation of a monomer to an existing polymer chain should also be made possible because the monomer comes into close contact, comes close together to that polymer. So mobility of the monomers as well as the polymers is a very important factor for the polymers to be able to extend their lengths. Now, in the dry phase, this kind of diffusive processes becomes less significant because the region dries up, right? But at the same time, if there are monomers which are close by, so that reactions can take place to extend the polymer, those kinds of concatenation reactions are favored in the dry phase. Whereas in the wet phase, you allow for a lot of mobility of the monomers, but at the same time, increase the chances that hydrolysis will break up the existing chains of polymers that were formed in the dry phase. So this cycling between the wet and dry phases due to this rehydration and dehydration cycles, which occur near the edge of volcanic pools, allows for periodic replenishment of monomers, which can then facilitate the extension of polymers in the dry phase, and so on. So we found, the reason this is useful is because such cycling can actually lead to an increase in the average length of the polymers produced. And you can see this from these two figures. The top figure shows you the average length of polymers in the absence of the rehydration-dehydration cycles, where the average length stabilizes to less than 10. So a polymer chain which is less than 10 monomers long. Whereas in the presence of this rehydration, dehydration cycle, even though the length of the chain also cycles between low and high values, the average length is much larger compared to the case without cycling. So this process of dehydration, rehydration across the period of a single day can lead to increase in average length, which in fact makes it more likely for functional biopolymers to be produced. So one of the studies that we carried out was to try and understand whether these primordial processes involving concatenation reactions and template-directed primer extension processes can lead to formation of long polymers which can take on complex structures. And even though we don't know, we could not really generate or say whether these complex long polymers could perform a catalytic function or some other important biological function, we can use the structural complexity and its similarity with the structural complexity seen in RNA enzymes like ribozymes to say whether such ribozymes can be synthesized spontaneously just through these concatenation and primer extension processes. So that was one of the main objectives of the work that I'm just going to talk about. And so what we did was to segregate the duration of a day into 3 distinct environmental phases: a morning to noon phase, which was essentially a dry phase, which facilitated this concatenation process, a semi-wet phase, which facilitates template-directed primer extension, and a wet phase at night, which is accompanied by rainfall and which increase in water content, which leads to hydrolysis of the existing RNA molecules that are already produced during the previous 2 phases. So since these are competing processes, we wanted to find out whether they can produce long structurally complex RNA molecules, which can be thought of as being capable of performing biological function like catalyzing reactions. So we developed a computational model where we modeled all these reactions. We divided the entire duration of a day into these 3 phases. And these 3 phases were distinguished by the kind of reactions they support: concatenation reactions occurring during the dry phase, template-directed primer extension processes occurring during the semi-wet phase, and hydrolysis, which breaks up the existing polymer chains occurring in the wet phase. And so we wanted to find out what were the relative durations of these 3 phases which would most favor the production of long structurally complex RNA molecules. And this figure shows you how change in duration of the dry and semi-wet phase affect the length of the RNA polymers that are produced, starting from a population of monomers only. So as we increase the duration of the dry phase, which means allowing more time for concatenation reactions to take place, we find that the length increases, and the length is in units of monomers. So 40 means a polymer of length 40 nucleotides has been formed, and so on. So as we increase the duration of the dry phase, we can see that the length of the polymers also increase because there's more time for polymers to extend through this concatenation process. Now, you may ask, why do we need both these processes which lead to increase in length, concatenation as well as template-directed primer extension? Well, we need both because concatenation is essentially a slow process, but we also need concatenation process in order to produce the templates on which the primer can attach and then be extended at a much faster rate. And so the combination of these two processes allow us to produce long polymers, which are long enough to fold in structurally complex forms, which by their similarity with ribozymes gives us some confidence to conclude that they can potentially act like functional biopolymers. So one of the key results that we found in this work was that increased duration, which leads to more concatenation reactions occurring, increases strand length, and some of these strands become templates which facilitate template-directed primer extension in the semi-wet phase. And increased duration of the semi-wet phase also increases the overall length. So if you can see that this is a heat map which shows how the maximum length, which is averaged over several days, change with the duration of the dry and semi-wet phases. So if you go along a row, you are changing the duration of the dry phase, keeping the duration of the semi-wet phase fixed. So increasing the duration of the dry phase then also implies that the duration of the wet phase decreases. And so there's less time for hydrolysis reactions to break up existing RNA polymers. And that is why for a fixed duration of the semi-wet phase, increasing the duration of the dry phase leads to increase in the maximum average length of these polymers. So you can see that we can attain lengths which are quite large, of the order of even 100. And some of these functional ribozymes are typically of the length ranging from 50 to 200. So even RNA polymers which are as short as 50 or 60 nucleotides long can fold in complex structures and are capable of catalyzing certain reactions. So the take-home message from this slide is a longer, so if you go along the column increasing the duration of the dry phase for a fixed duration of the wet phase, the length increases. If you go along, if you go along a row, if you go along a column, increasing the duration of the semi-wet phase for a fixed duration of the dry phase also leads in increasing the length of polymers, although not as significantly as in the previous case. So why does it help to increase the duration of the semi-wet phase? Because increasing that duration allows primer extensions to occur for a longer period of time. So if you have a large template, which was initially, to which a short primer initially attached, and we want that primer to be extended completely, and form at least an RNA molecule which is as large as the template. We need enough time for the template-directed primer extension processes to take place for the primer to be fully extended and form a polymer which is as long as the template. Now, increasing the duration of the semi-wet phase facilitates that process, but it also serves another purpose. And that purpose is to reduce the size of the dangling ends, which are more prone to hydrolysis during the wet phase. So if you recall the figure that I showed earlier here about how hydrolysis acts preferentially on bonds which are unprotected. So if you look at the figure on top, for instance, you have a template here, and you have the primer which is being extended from right to left. And the larger the amount of time you have to increase the length of the primer, the smaller will be the length of the dangling portion of the template, and less prone it will be to hydrolysis reactions which can break it up. So these two processes can work in conjunction, to increase the length of the RNA strands in such a way that it allows for them to fold into complex shapes. So why do RNA strands fold? Because it is energetically favorable for them for long RNA strands to fold. But folding depends on the sequence, and one can calculate folding free energies associated with different RNA sequences to figure out how likely a given sequence is to fold into complex structures. So what we did was to do that calculation. So we were able to extract from our simulations all RNA sequences, fold them using an algorithm that was previously developed, and found that the RNA sequences that are generated in the simulations fall into 4 distinct categories, which are distinguished by the kind of structures that they produce. And this figure shows the 4 different structures and how abundant they are, and how their abundance depends on the duration of the dry and semi-wet phases. So you can see that complex folded structures start forming even for low durations of dry and semi-wet phases, because these are, even though they are relatively shorter RNA polymers, they are long enough to fold into these kind of structures which have an internal loop as well as an external loop here. Now, these structures are interesting, but they are not functional in the sense that they are not similar to any of the known structures of functional ribozymes which have been discovered. But these are the ones which dominate because they can form for even lower durations of dry and semi-wet phases, but they can also form because even if the polymer lengths are not so large. But the ones that we are interested in are actually structures which are similar to the ones that are found in existing ribozymes, which have catalytic properties. And these are the structures which are formed only for certain durations of the dry and semi-wet phase, and that too with fairly low abundance. So if you can see the figures. I don't know how legible they are. So for a duration of the dry phase of 8 hours and semi-wet phase of 8 hours, roughly 0.1% of the structures generated are of this type. This is called a hammerhead structure, and because it's similar to the structure of a so-called hammerhead ribozyme. And so these structures, even though they are not as abundant as the more frequent structures shown above, they are still produced, but they are produced in greater numbers the greater the duration of the dry and semi-wet phases are. And on the right, here you can see a clover leaf kind of structure, which is typical of a transfer RNA molecule, which plays a very important role in protein synthesis. And these kinds of structures also require RNA polymers of length approximately between 70 and 100 to form. And as you can see, they are also formed preferentially for relatively longer duration of dry and semi-wet phases. Okay, so the take-home message that I want you to get from this is that just non-enzymatic concatenation and template-directed primer extension processes can be sufficient to produce long enough RNAs which can fold into complex structures which can be thought of as being proxies for biological function. And some of the RNA strands produced fold into structures that are very similar to the structures of existing ribozymes, which are RNA catalysts. And so this gives us hope, at least it can be thought of as a proof of concept, that basic processes like concatenation and template-directed primer extension can lead to creation of RNA enzymes. So this addresses one of the problems associated with this RNA world hypothesis. How do we get RNA enzymes which can then speed up reactions like the replication of RNA sequences and so on, and really kickstart the RNA world and lead eventually to the formation of the first living organism, even though it's completely based on RNA. So coming back to the original question that we posed some slides ago, we were able to address this question of how the first ribozymes emerge in the absence of catalysts. But it is not enough to address this question in order to understand whether RNA world is a viable hypothesis for the origin of life, whether life really existed in the form of RNA which could perform all three of the functions that are now performed by DNA and proteins in our current world. So it's one thing to say that these components can be synthesized. So we've seen that the basic building blocks like RNA nucleotides A, U, G, C can be synthesized. It has been done recently in the last few years by two different groups, one in Germany and one in the UK. We know that RNA polymers can form non-enzymatically either through concatenation processes or through template-directed primer extension processes or both. And by combining both these processes, we can produce RNAs which have complex structures which are similar to the structures of existing RNA enzymes. So this gives us hope that these individual components which were essential for the RNA world to be a plausible hypothesis for the origin of life can be formed through non-enzymatic processes. But there's one thing missing, and that missing thing is a compartment. So we need a compartment, and compartments are typically formed of lipid vesicles. But there's another problem with this RNA world scenario which has not been addressed as yet, which still remains an open question, which is how do these components come together even if lipids exist, and even if they encapsulate RNA strands and templates and ribozymes and nucleotides and so on. How do you go from a collection of components to a living organism which can replicate and function in this self-sustained manner, and which can also pass on hereditary information to its progeny? So how is life possible given that these components can be produced? So that remains an open question, which I'm going to address in the next slide. So this is ongoing work, which is not yet complete, but it gives some insight into how the system can evolve towards increasing complexity through functional diversification. And so this is how we are thinking about it. So suppose you have a protocell which is made up of a lipid vesicle, and inside these protocells are these different RNA components, including nucleotides, which are necessary to increase the length of RNA strands and so on, and perhaps some RNA enzymes as well, which can carry out certain biological functions like replication. So suppose you have this system which is now enclosed in a lipid vesicle, which we call a protocell. How do we go from there to a protocell which can replicate, and inside, and not only replicate, it can transfer information from itself to its progeny in an error-prone manner, which allows for new variants to be produced, which eventually allows for better protocells with more functional diversification of the internal components, and so on. So how do we kickstart evolution starting from a protocell which encloses some of these components that are the key components of an RNA world, right? So we started addressing this problem by thinking that because of this, some of these replication processes that I talked about, like the template-directed replication process, you can produce enzymes, right? But the appearance of the first enzyme was a chance event. It so happened that a certain sequence was produced, which was long enough and could fold into a sufficiently complex structure that allowed it, that imbued it with a certain functional capability. And let's assume that the functional capability was its ability to speed up the replication process of other RNA molecules. So the first enzyme may have appeared by chance, but if it is able to speed up the process of replication of other RNA molecules, it can lead to a diverse population of RNA molecules. And within that diverse population, some molecules would emerge with a somewhat different function than the replicase, which was capable of speeding up the replication process. And so in a population of protocells, you may have some cells in which you can have a few more of these ribozyme-like molecules, each of which can perform different types of functions. And the question is whether evolution of this population of protocells, some of which contains some RNA components, maybe not even a single ribozymes, some which contain one ribozyme, some which contain two ribozymes, some which contain three ribozymes, and so on, how the evolution of this population will eventually lead to a population of protocells in which the protocell which has multiple ribozymes which can perform different biological functions dominate in frequency. And so this is what we found based on preliminary results. So we had this model, and I'm not going into the details of this model, of how you can produce these ribozymes with a small probability, and then track the evolution of the protocell population to see if the protocells which have a larger number of functionally diverse ribozyme increase in frequency at the expense of protocells which has fewer ribozymes or even no ribozymes at all. And we found out that if you have the probability of formation of a certain ribozyme to be above a given threshold, it can act in this, it can create a positive feedback which leads to production of other ribozymes and so on, which then leads to a population of protocells which are dominated by ones which have multiple ribozymes performing different functions. So this is what we are looking for in order to address how evolution, starting from simpler types of protocells, led to increasing complexity through increase in the number of ribozymes which could perform different types of functions. Okay, so how am I doing on time? I think I've almost,
Yes, sir.
moved towards the end. Okay.
Yes,
So
sir.
let me stop by just giving you a glimpse of the other aspect of the problem. So far I've been talking about how one can figure out whether the RNA World Hypothesis was a plausible one by trying to understand how the components of the RNA world can be produced non-enzymatically through standard chemical processes like concatenation and template-directed primer extension. And secondly, how the RNA world can be kick-started in the sense of producing a living protocell which is capable of replicating and functioning in an autonomous manner through this process of evolution from a population of protocells, some of which would contain a small number of ribozymes to begin with, but which would later evolve to a population where most of the protocells would contain a large number of ribozymes with the capability of performing different functions. So that's the first part of the story of origin of life. The second part has to do with the transition from the RNA world to a DNA protein world that we now see prevalent in all living organisms that we are familiar with today. And that world has, the transition was facilitated by the development of the language of genes. So what do I mean by that? We start with a genetic alphabet, but the genetic alphabet, but the system has to figure out a way of encoding information using the genetic alphabet in such a way that the information can be subsequently decoded into small chunks, which would then be used to produce autonomous self-replicating entities that we call life. And that happens when you can figure out a way of decoding that information, and that requires development of a language of genes, and it requires the development of what's called the genetic code, which allows you to take a sequence and from that sequence figure out what sort of proteins can be synthesized. And that is only possible because of the mapping between the triplet of nucleotides called codons and amino acids. So one of the key questions associated with this transition from an RNA world to a DNA protein world is the question of how this pattern of association between codons and amino acids emerged. Okay, so I'm not going to talk about this, and I'm going to skip to the last slide of my talk. Which makes me come back to the question of what do we know about the origin of life? We still don't know much. And I think of it as trying to build an extremely complicated jigsaw puzzle with lots of pieces. We know bits and pieces of the puzzle. We can understand how parts of it work, but we still don't have the full picture. But the puzzle is enticing enough to motivate us to keep trying to figure out the answer to the question of the origin of life. So thank you for your attention, and sorry for overshooting the time by a few minutes. I want to stop by acknowledging my students and collaborators who were on this journey and involved very much with this, with all the work that I talked about. So thank you.
Thanks a lot, sir. That was a great talk. And now we can take up some questions if someone has.
Sure.
Yes, sir. So if you're comfortable, you may turn on your microphone and ask the questions, or you may write it down in the chat box. Either way, it's fine.
Sir,
Yeah.
so when we were talking about the phases, the wet phase, semi-wet phase, and dry phase, in one of those diagrams, I noticed that there was no maximum length of given for 12 hours dry phase and 10 hours semi-wet phase, and 10 hours dry phase and 12 hours semi-wet phase. So I was wondering if 2 hours of uh, a wet phase is not enough for the hydrolysis to provide monomers to replenish the total number of monomers for the process to continue.
Yeah. So let me go back to the slide. Is this the slide that you were talking about?
Yes, sir.
Okay. And your question is, we have, so the maximum we have for the dry and semi-wet phase duration is 12 plus 8, 20. So you still have a wet phase duration of 4 hours.
Yes, sir.
Yeah. So the question is whether that is sufficient enough to replenish the pool with enough monomers to continue this process of elongation of the polymers when the next dry phase starts.
Yes, sir.
Right? Well, we don't know for sure. The point is that if you so what we find is if you have such a long duration of dry and semi-wet phases, it leads to large sequences, assuming that monomers are available. If monomers are exhausted, of course, this whole explanation breaks down. And then we can no longer claim that these kinds of long RNA sequences will be produced. So this
So
is
4 hours is
more of a proof of concept, right? So we know very little about the environmental conditions that existed about 4 billion years ago, and the ones which eventually gave rise to the first living organism. We can only speculate. And this leads to some sort of exploration of the possible scenarios which can yield results that are supportive of this hypothesis that it's possible to produce long RNA molecules with enzymatic activity. So
yes,
the answer
sir.
to your question is if they are not, if they are, so we assumed that monomers are unlimited, and that's why there was no point at which we had monomer deficiency, which stopped these extension reactions. But if monomers are not unlimited in that small region, then of course it will depend on the rehydration phase to replenish it, and then the results can change.
Okay, sir. Okay, sir. Sir, I had one more question.
Yeah.
Sir, you said that after a certain threshold value of ribozymes is reached, then uh, the lipid compartments with more ribozymes are preferred, like their percentage in the total lipid compartments increases. Sir, how does that happen? Because the lipid compartments are still not reproducing, I think.
Yeah. So let me come to that. So I didn't say that. Can you repeat the question again? I'm sorry, I may have misunderstood it.
Sir, you said that the number of lipid compartments which have ribozymes, which have more ribozymes, increases after a certain time. Like they are preferred over those compartments that do not have ribozymes. So, how come that happens? Because
only under certain conditions, and through the process of evolution, right? You can see that you have these protocells with different number of RNA molecules, some of which are ribozymes. So my claim was, okay, let me tell you a bit about how we are doing these simulations. So we take a protocell which contains these components and a population of such protocells, and then we carry out all the reactions, different types of reactions within the protocell that can produce new types of ribozymes, right? So once in a while, due to these reactions, it can not just produce, it can produce, structurally complex RNA molecules, some of which has a probability of acting as a ribozyme, right? And once in a while, one of these molecules will be produced which acts as a ribozyme. So you can have ribozymes produced inside the protocells, different types of ribozymes produced with certain probabilities which have been specified. So for instance, in this example, there's this probability of formation of nucleotide synthase. So this is an enzyme which speeds up the synthesis of nucleotides, the monomers A, U, G, and C, right, which are essential for polymer extension. And so you can have such processes which leads to these ribozymes being produced, but then the question is, why is it that protocells which have more of this will be preferentially selected, right? Well, the answer is it doesn't always get preferentially selected. There are certain conditions which have to be met for those protocells which contain a large number of functionally diverse ribozymes to get preferentially selected. And those conditions are determined by the probability with which these are produced, as well as the way we are implementing the evolution process, the way we are updating the population of protocells. So when the protocells reach a certain volume, which is determined by the number of components that's present inside it, it splits up into two protocells. And in order to keep the population fixed, an existing protocell from that population is removed. But the one which is removed is the one which has the smallest number of components. So this is something that we put in by hand and is justified because protocells which have smaller components and therefore smaller volumes are less likely to contain functional material which could speed up reaction processes inside those protocells, and they would have a selective disadvantage relative to protocells which have more functionally diverse molecules. So the selective advantage associated with larger number and more functionally diverse molecules leads to the evolution of the population under certain conditions in a direction that prefers protocells which have these characteristics. It's not automatically decided.
Sir, you said the protocells divide. I mean, is it do the ribozymes help in that? Ribozymes help in the division of protocells.
No, in our model, the ribozymes don't directly help in the division of protocells. What we assume is if the protocells reach a certain threshold volume, and volume here is measured simply by the number of components that are inside the protocell. If the protocells cross a certain number of components, it divides into two, and then the components are randomly distributed among the two daughter cells.
Okay, sir. Thank you, sir.
Sir, there is a question in the chat box which
Yeah.
is like, so Taq polymerase is same as dry phase.
I'm
This is the
not sure
question.
I understand that question though.
Sir, I guess he meant like as in the dry phase, the RNA molecules are joining. The Taq polymerase and PCR functions in a similar way to elongate the chain. So, is it the same enzyme which is used in dry phase? I guess that is what he meant.
No, we are not using any enzymes, right? I mean, the whole point is to carry out these reactions without enzymes. These are standard reactions which occur at specific rates, which are the rates that have been measured in the lab. And all these reactions are spontaneous reactions which are not sped up by enzymes. There were no enzymes to begin with. So we cannot invoke enzymes right at the beginning, because then the question will be, where did the enzymes come from? So the question that we are asking is, enzymes have certain characteristics, right? They have to be functional biomolecules. Functional in the sense that they are structurally complex enough to be able to perform a certain biological function like cleavage or replication and so on. And by definition, such structurally complex molecules have to be long polymers. But we are trying to answer the question, how did the long polymers emerge in the first place without enzymes? We cannot use enzymes to produce these polymers because there were no enzymes to begin with.
Yes, sir. So Varun, if this was your explanation, or is the question a bit different?
It was this only.
Okay, thank you. Sir, I have a question.
Sure.
So, like in the primitive environment, in the early stages of Earth, it was kind of like different chemical composition and different temperatures. So, during these experiments, did you take that into account, or is it a different experiment which cannot be related to this?
Different. You have to make the question a bit more precise.
So, like
By different chemical components.
like the primitive environment, the atmosphere was a different chemical composition compared to the recent chemical composition of our environment, the atmosphere, right? So,
Right.
like, so that might affect the reactions which are going on at that time, and the reactions which we perform in the lab during this time. Like there might be a difference. So, are you containing that kind of environment, or is it the same as this environment?
No, so the rates, the experiments which determine the rates of these processes, which we used for our modeling, are all in vitro vitro experiments. Yeah, I mean they are just done in the normal environmental conditions of the lab of the current situation. And I'm not, I get your question in the sense that you are probably have something in mind which is related to the Miller-Urey experiment, where they passed where they tried to simulate an early environment by filling a flask with various chemicals like oxygen, nitrogen, carbon dioxide, methane, and passing electrical discharge, and found that they were able to produce some amino acids. So whether those kinds of environmental conditions related to the percentage of different gases like methane and carbon dioxide and oxygen and so on, they don't affect the results much. But your point is well taken, because what is important is what we took into account is the fact that a lot of these rates are measured in situations where there are mineral-rich environments. So these are template-directed processes, though are taking place on the surface. So the surface is mineral-rich. And if the surface is mineral-rich, it has been shown, again by experiments done by geochemists, that certain reaction rates, especially that of primer extension processes which are directed by the template, increase several fold. And yes, so we did take into account the fact that these environmental conditions are such that they favor the template-directed primer extension processes, especially in the presence of a mineral-rich environment. So we did not, we don't consider the gaseous environment, because I don't think it matters much here as far as these reactions are concerned.
So because I was thinking, like, if the moisture content in the primitive environment would be too high, so the H2O molecules in the atmosphere might affect the RNA nucleotides reaction. Like they might prevent some reactions due to hydrogen bonding also, if the moisture content was too high.
Yeah, well, it is possible, but then we are looking at a situation where these reactions are taking place close to a volcanic pool, right? And so there's a lot of the temperatures are very high there, and because of the high temperatures, it can probably, I'm not an expert in the environmental geochemistry of primordial Earth, but the fact that they are taking place close to volcanic pools means that we are dealing with pretty high temperatures, and which would address the question of moisture content possibly to some extent. Right?
Yes, sir. Yes, sir. Thanks a lot, sir. Are there any more questions in the chat? So if there are no more questions, then thanks a lot, sir, for this amazing talk. And we are looking forward for more such interactive sessions in the coming future.
Thank you.
Thanks a lot, sir. It was a great talk.
Thank you.
Thanks a lot, sir.
Bye.
Bye.
Thank you, sir.
Thank you, sir.
Hello?